In this week's lab, we're going to pretend to be biochemists who've just purified a protein. And before we go to the next step, before we start doing experiments with this purified protein, we need to know what the concentration of that protein is. And so there's several ways to do this. We're going to talk about, and you're going to basically do one of those ways using something called the Biuret assay. Now, the Biuret assay is an indirect assay. And by indirect, what I mean is we're counting on a particular property of the protein to allow us to indirectly figure out how much there is. So how does this work? Well, the Biuret assay is a chemical that you're going to add to a portion of your protein sample. The Biuret assay the Biuret solution itself interacts with the peptide bonds of that protein, and when it does so, it turns purple. So, the more protein you have means the more peptide bonds, means the more interactions with the Biuret solution, means more purple. You see how that's indirect there? So if we were to do that, and what we see here is if we have no protein here, the Biuret assay, the Biuret solution stays this sort of light blue color. But with the more protein we have, the more purple it's going to get. So if we had a way to quantitate the color change, to quantitate just how purple that solution became, we can then use that to quantitate how much protein we have. We can. It turns out that that purple color, when Biuret reagent turns purple, it starts to absorb more light at 520 nanometers. Notice it absorbs 520, which is in the green spectrum, but it's purple. Well, that's because that it absorbs the green light and transmits the sort of purple wavelengths of light. That's why something is the color it is, because it allows through or reflects one color while absorbing all of the others. So purple Biuret reagent absorbs very well 520 nanometers of wavelength of light, whereas the blue form does not absorb that particular wavelength very well. All right, great. So now we've got this property of the purple Biuret solution. It absorbs very well 520 nanometer wavelengths of light, whereas the blue form does not. Okay, if only there was a way to actually measure and quantitate absorbance. Well, turns out there is. Spectrophotometers. Now, you watched the video on using a spectrophotometer before coming into lab. Well, here is the theory behind spectrophotometers. The idea being that within this machine, within the spectrophotometer, there is a light source that is passed through a prism which separates the different wavelengths of light, which you then set the machine to allow a particular wavelength through. That particular wavelength goes through your sample of interest, and then the amount that has been absorbed is detected and displayed on a readout. So you can take your spectrophotometer, you can set it to 520 nanometers and reading absorbance. You can put your cuvette there that you have your sample of protein that you've mixed with your Biuret solution, and it's turned purple. And now we're going to measure the absorbance of just how purple it's become, just how much of this 520 nanometer wavelength of light it is absorbing. And you'll get a reading. You'll get an absorbance. Now that absorbance is unitless. It'll be like 0.2 or 0.5 or 0.7, no unit. And so the question is, well, that's absorbance, but we want to know concentration. So how is it that we convert the absorbance that we get from the spectrophotometer to concentration, milligrams per mil? And the answer is we have to build a standard curve using standard proteins. What's a standard protein? Well, a standard protein is a series of protein concentrations that we already know. In this lab, we're going to use the protein albumin, and we're going to use 1, 2, 3, 4, 5 different concentrations of the albumin protein, 0.25, 0.5, 1, 1.5, and 2. And then what we're going to do is you add Biuret solution to each one of these known concentrations of protein. You allow that to sit and become purple, and then you put those into the spectrophotometer and you get a reading in absorbance for each one of those. This is the data that you are going to generate that will allow you to make a standard curve. Because once you get those absorbance readings, you have right? You've got the ability to plot this information. So a data point would be 0.25 milligrams per mil, and the absorbance of, let's say, 0.2, and so on and so forth. So you're going to generate a bunch of data points, and then you're going to use those data points to plot out onto a graph using either Google, what's that Google program? Google Notes, Google Numbers, one of the Google programs, or you can use Excel. And so you just plot that in. Okay, well, let's see. I had the 1 milligram per mil. It absorbed 0.1, so we'll put a dot there. Our 4 milligrams per mil was about 0.2. Put a dot there. And when you do that, you're going to get a line. If you do it carefully, you'll get a line. Now, that line that you produce is your standard curve. And now there's two ways you can use your standard curve in order to determine the concentration of protein in your sample. Both of them rely on taking that protein sample that you've added Biuret to it and popping it into the spectrophotometer and getting an absorbance. Now that absorbance, you can just say, oh, okay, well, it was 0.5 was the absorbance. And then you can eyeball it. You can say, okay, 0.5, we go over, we'll go over until we hit our standard curve. Then we go down, and it looks like it's about 6.75 milligrams per mil. Now that's eyeballing it. The better way to do that is this line has an equation to it. And you can get that equation to the line from Excel, right, when you build that graph in the first place. And that it'll have the form y equals mx plus b, where y is your absorbance, x is the milligrams per mil of proteins. You know your absorbance of your unknown protein sample, so you would plug that in for y. You would then solve for x, and that would give you your the concentration of your protein sample much more accurately than just eyeballing it. Okay, so that is what you are going to do. We are giving you the absorbance at 520 of all of your standard curve of your standard proteins. You'll build the standard curve, and then we will give you the absorbance of your unknown protein, and you can use that to solve for X, which is the milligrams per mil of your protein.